This talk is about the Medical Sciences and Engineering programme. This is for both versions of the programme, both the BSc and the MSci. So by way of introduction, my name is Dr. Darren Player. I'm one of the programme directors, and I'm also the divisional undergraduate tutor. So just to give you an introduction to UCL, UCL was established in 1826. It was the first university to admit students independent of gender, of religion or cultural background. It has 33 Nobel Prize winners, is ranked #1 in London for medicine, and is a top 10 faculty of medical sciences worldwide. As you can see below, there are a couple of images here of UCL back in the Victorian time, so in the 19th century, and also in the present day, and really it hasn't changed very much at all. So just to give you some context to the programme, so the programme was developed really to meet a particular need of the healthcare sector in the UK. So as you can see here, the medical technology sector is rapidly growing. So there's a large increase in the number of companies that have been established within this particular area of healthcare. The growth in medical technologies has actually outgrown some of the traditional pharmaceuticals. So what's really important now is that we can develop graduates who have skills to meet the particular demand in this area. So this is of national strategic importance and also of international importance, that the medical technology industry needs innovation for continued success. And we need graduates who have got the skills to meet that particular demand. At the centre of this, research needs to be clinically informed, so it needs to be driven by the needs of the patients, and we need to be developing engineering solutions for global health challenges. So this degree is a cross-faculty degree. The home division is the Division of Surgery and Interventional Science. This division has 3 departments, one in the Bloomsbury campus, one at the Royal Free Hospital in Hampstead, and one at the Royal National Orthopaedic Hospital in Stanmore. The degree is also shared with UCL Engineering. The degree is also run in partnership with the UCL partner hospitals across London. So our vision as a programme is to support teaching and research and address the challenges of disease, ageing, mobility and independent living. The importance here is that we bridge the gap between the 2 disciplines, so engineering and medical sciences, with the hospital settings, so that we really can develop these clinically informed solutions. There is a partnership between the world-class academics and facilities at UCL and the clinical experts, and that's really needed to speed up the development of new treatments. And we've seen this with the latest developments related to COVID-19, and one of my colleagues, Dr. Zara Mori, will touch on this a little bit later. There's also developments in diagnostics, prevention strategies, and research breakthroughs related to translational science with the importance of clinical excellence for patients. So the important thing here is that everything has to be clinically informed, and that the 2 disciplines of engineering and medical science need to come together to develop those solutions. So the main aim of the programme is to investigate the causes of human disease and understand how to develop innovative therapies and technologies to improve the quality of life of these patients. So I'm now going to give you an overview of the programme. So throughout the programme, modules are taken in both the Faculty of Medical Sciences and the Faculty of Engineering Science. So in year 1, modules are taken in the area of kidneys, hormonal control of human physiology, fluid balance and nutrition, the gut, liver and drug metabolism, and cardiovascular and respiratory function, health and disease. And in the Faculty of Engineering Science, mathematics and modelling, medical instrumentation and materials and mechanics. Now throughout the programme as well, there's modules that sit across the 2 faculties. So in the first year, there's an introductory module, which starts at the very first 4 weeks of the programme when you enrol, which is an introduction to engineering, biology and medicine. There's also continuous design and professional skills modules and professional practice modules, which run throughout the degree. And I'll touch on that a little bit more detail later on. So there are 8 compulsory modules that sit across the 2 faculties. In year 2, in the Faculty of Medical Science, you study the functional nervous system and brain. musculoskeletal systems in health and disease, and infection, inflammation and repair. And in the Faculty of Engineering Science, continuation of mathematical modelling and analysis, manufacturing and regenerative medicines, fundamentals of biomechanics and medical instrumentation 2. And there's also a professional medical practice module, which is a continuation of the professional practice module or design and professional skills from the first year. So in year 3, you will actually choose a particular stream, either within medical sciences or in engineering, and that's a bespoke stream of modules that fit within those two disciplines. You'll also undertake a compulsory module in professional clinical practice, where you have the opportunity to undertake short placements in hospital to understand what the clinical challenges really are. In year 3, you'll also undertake a research project, where you'll have a chance to engage with the world-class research that is happening at UCL. So if you're registered for the BSc, at this stage you would exit at the end of year 3. If you're registered on the MSci programme, you can continue into the 4th year, where again you take modules that are streamed in either medical sciences or engineering, and you undertake a further research project. And at the end of the year 4, you would exit with an MSci. Now, what's important to communicate here is that the programs are set up as cognate programs, which means that throughout the course, you can interchange between the BSc and the MSci. There are some conditions to this, so if you want further information, we can give that to you. So just to highlight the professional practice modules, as I mentioned, in year 1, there's design and professional skills. In year 2, there's professional medical practice. And in year 3, there's professional clinical practice. Now, the purpose of these modules really is to connect the academic learning with key skills such as teamwork, project management, presentation skills, creativity, innovation and enterprise, and leadership. And what we really want is students to gain and develop a range of skills that you can then apply in professional careers. So we adopt an innovative teaching style, a blended learning approach, where we embrace digital media and technology. So for some of the teaching content, we use the flipped classroom idea. So a traditional learning environment would be students come into a lecture theatre, you would listen to the lecture, you would take notes, you would then go away and you'd do some activities at home. With a flipped classroom, you would actually undertake a lot of the lecture material online through the virtual learning environment, and then you would come in for classroom-based activities. The idea here is that you would understand the foundational knowledge in the lecture that you would go through in the virtual learning environment, and then you would come into the classroom where you'd be able to discuss those ideas at a higher level and start to challenge you to think in a different way. So there are short video lectures which are part of this series, and also there's an opportunity, for example, if you don't understand anything, you can obviously pause the video, you can go away, you can read some of the other additional material that might be a part of that session, and then you can come back when you've actually understood maybe what you haven't understood previously. So in-class time really is then devoted to exercises, start doing some more complex tasks, projects and discussions that are related to that foundational material. So this is an example of the virtual learning environment. So what you can see here is a lecture that's being delivered particularly about liver disease. Now what you can see is that it's not just a simple recording of a lecture, it very much is interactive. So you can interact with some of the content there. There are questions that are posed that you can answer. And there's also an opportunity to engage in student forums where you can ask students and staff questions. So in terms of the assessment, there's a variety of assessments that we use as part of the programme. And the idea here is that we're wanting you to develop a broad range of skills and assess you accordingly. So it's a blended approach between multiple choice questions, short essay questions, written assignments and laboratory reports, and presentations and group work. So in terms of graduate careers, where do we expect students actually to work once they've graduated from this programme? So students will be capable of working alongside clinicians to deliver this increasingly technology-driven standard of care. So for example, you can see here in the image, This is an example of a surgical robot, and we would expect students to have the skills and understanding to work alongside clinicians to develop technologies such as this. So students will have a new skill set that they'll be able to take to the graduate market, and you'll be able to work with a lot of these companies that are developing in this area. So we'd expect students to find employment within a team, for example, of surgeons, cell biologists, engineers, material scientists, mathematicians, modelers, nanotechnologists, and also working on the regulatory side to actually get some of these technologies into the clinic. So just to go over the admission criteria. So an A is required in both mathematics and biology, and then a B is required in either physics or chemistry. If you are undertaking an international qualification and you need further clarification on the admissions criteria, please contact us and we'll be able to help you with that. So at UCL, we have a number of opportunities for student support. So we have student mentoring. There's transition mentors, where students who are already part of the program will actually mentor the first year students as they transition into university life. There are academic personal tutors, so you'll all be assigned a personal tutor at the start of your degree program, and you will meet that academic personal tutor regularly, where you can talk through not only just academic issues, but pastoral issues as well. There's student support and wellbeing services at UCL, and as part of that, wellbeing and mental health. There's disability support and also counselling services. So thank you very much for listening. There are going to be some other talks related to research, so we hope that you enjoy these talks that are coming up. Thank you very much.
Hello, and welcome to my talk that is part of the Medical Sciences and Engineering degree program here at University College London. My name is Zahra Mohri, and I'm a lead for a number of modules in Medical Sciences and Engineering. My colleague, Dr. Dan Player, has a presentation discussing the program itself, and you can hear about the research of another colleague, Dr. Alvina Qureshi, which is very multidisciplinary in nature, in another talk. But in here, I want to discuss something different. I'm going to take you through a journey that has affected us all in the UK and across the world, through something that we can all relate to. You have guessed it right. I'm going to talk about the COVID-19 and how UCL academics have been working hard in trying to combat the virus. Here in UCL, we have a large number of COVID-19 experts who have mobilized resources to focus on answering a number of questions. For example, trying to find a cure by sequencing the virus genome and ultimately designing a suitable form of vaccination. By improving the diagnostic aspects of the battle against the virus, as well as providing advice for the leaders within the UK and other countries in the world. In addition, these dedicated members of our community are providing expert comment through media and their publications with regard to the economical impact of the virus, the political issues, as well as mental health and emotional well-being. You can read more about their work that is fascinating and wonderful in the address at the bottom of this slide. The topic I have chosen to discuss is very close to medical sciences and engineering. It's UCL Ventura. UCL Ventura brings together a group of engineers, the leaders in the field with their teams, to work side by side with a group of clinicians and their group. They all collaborate with industrial partners, trying to design and manufacture a breathing aid for critically ill patients affected with the virus. And the wonderful news is that they have made a discovery. They have designed a device they call CPAP, continuous positive airway pressure. The discovery was very rapid within a very short period of time. In less than 100 hours from their initial meeting to the delivery of the device to the market. CPAP has passed regulatory approval, has been tested successfully in a large number of patients in various hospitals of UCL. And during this time of global pandemic, UCL Ventura team are sharing their design, the details of their design and manufacturing with the relevant groups in different countries across the world. How it works is basically pushing oxygen and air into the mouth and nose under continuous pressure. This allows the airways to remain open and increases the entry of the oxygen into the blood circulation. And indeed, it has proved successful in helping the very critically ill COVID-19 sufferers, which means that the clinicians then do not need to use invasive ventilation technology. I want to take you back to medical sciences and engineering now. The various modules we teach our students, some of them are very relevant to the COVID-19 story. For instance, one of them called Cardiovascular and Respiratory Function in Health and Disease that I lead teaches our students about the heart and the lung, their anatomy, their function, how the respiratory apparatus works. And as part of this module, they have a practical session where they use an spirometer to measure their breathing under various conditions, plot their data, and analyze it. Another module that is taught by my colleagues in the engineering faculty is called medical instrumentation. During this module, they learn how to design devices. And for instance, we have an image of their work trying to build an electrocardiogram, ECG. The design continues through the second and the third year. And I must acknowledge our second year student, Noor Zaini, for sharing these images with me. So within the second year, we have a number of modules taught by the engineering faculty, where they're still learning how to use mathematical modelings and using designs to help the healthcare profession. From the medical sciences, one of the modules that, again, I lead is called Infection, Inflammation, and Repair. And as the name suggests, the students learn about our immune system. How do the molecules and the cells within the immune system work to fight against an invading pathogen, be it a virus, a microbe, a fungi? So with the current crisis and the situation of the lockdown, we are reading, all of us, pretty much about the COVID-19 discoveries. We're hearing about how it's important to keep the immune system healthy through exercise and healthy diet. And our students can understand the scientific terminologies used in those publications and in those papers. So to summarize, UCL community is in the front line within UK and the world to help the fight against the virus. And they are making a lot of progress. And medical sciences and engineering is indeed a very unique degree. It brings the two faculties together, it merges them together. Our graduates will be equipped with a specific set of skills that will help them in their future career. Thank you for listening. And if you are a sixth form student, I wish you all the best in choosing your future higher education path.
Good afternoon, everyone. I'm Dr. Alvina Qureshi. I'm one of the program directors of the Medical Science and Engineering program, and I'm going to give you a mini lecture on some research that I've carried out here at UCL on building artificial tissues for human transplantation. And the reason I wanted to share this with you is because it's a great example of how a multidisciplinary team have worked together to engineer a solution to tackle a real-life healthcare problem. And this theme is something that you will discover more about and learn about when you join our program. So what is tissue engineering to start with? Well, it's basically building or fabricating functional 3D tissues outside of the body, so in a dish. And these can be tuned in size, shape, and function to address the required needs. So depending on which tissue or which organ we are engineering, we're aiming to replicate, we will tune those properties accordingly. And so to do this, we need three key things. We need a material or a scaffold in which to start building the artificial tissue. We also need cells that would populate this tissue. So these can be obtained from deceased donors, so from the tissue of interest. And these are then grown together onto the scaffold in giving the right growth factors, the correct nutrients. And so these are often grown in a bioreactor where we can control all of these different parameters. So there's a variety of scaffolds that we could choose depending on the tissue of interest. Different materials have different advantages. So some might be stronger, some might be more porous. So ceramics, for instance, are a popular choice for bone tissue engineering because they are porous and replicate the structure more similarly to bone. Natural polymers are something that I've used, like collagen, because collagen is the most abundant structural protein in the human body, and therefore it's a good building block to begin to start to build our artificial tissues from, because the body is more likely to accept these tissues, so it's more biocompatible. So then we also need cells to populate on these scaffolds. So stem cells are a cell choice that are often quite popular because they hold great therapeutic potential. The properties of stem cells, so we can see that they can divide and self-renew, so they can replenish their own stem cell population, but they can also differentiate and divide into different specialized cells. So if they're in the right environment or they're given the correct cues, they can grow to be nerve cells, blood cells, muscle cells, and so on. And stem cells in the body, their function is to repair and replace damaged cells and tissues. So this is why they are a popular choice for our artificial tissues too. So my area of research was the cornea. And why is this important? Well, the cornea is the outermost transparent surface of your eye, and it acts as our window to the world. So because the fact that it's transparent, it allows light to pass through to the back of the eye, to the retina. And this is what enables our normal vision. And so the cornea is made up of different layers. There's 5 different layers. And as tissue engineers, we aim to replicate each of those individual layers. These are affected by different diseases, maybe different injuries. So we target each of those layers separately. I'm going to focus more on the stroma in today's talk. And this is really to address the healthcare problem that I was telling you about, and to kind of tackle this unmet clinical need. So we know that across the world, there are 10 million people currently that are suffering from corneal blindness. And this can occur through either disease, through heritable conditions, or through injury, such as acid or alkali burns. And as you can see from the pictures, what this leads to is a very damaged cornea. So you can see here there's a lot of opacity, we have vascularization, so there's blood vessels that are infiltrating from nearby tissues. And this causes the loss of vision in these patients. And so typically they would need to receive a transplant, but there is limited donor tissue supply. So just to put it into context, in the UK, to meet the demand, we need 70 corneas a week. But the donors that are there, 88% of organ donors are still not willing to donate their eyes. So we really need to find alternative to try and improve the understanding of, what are the differences between healthy and disease cells, so that we can try and improve our treatments. But also, rather than using one donor tissue to treat one patient, we can try and develop alternative treatment strategies so that one cornea could treat multiple patients, and therefore we would need fewer donor tissues. So let's look at the corneal structure in a little bit more detail. You can see here a diagram of the corneal stroma, and this is made up of sheets of very aligned, highly aligned collagen fibrils that are arranged in a basket weave pattern. And these fibrils have spacing between them, and the spacing is maintained with these glycosaminoglycans. And it's this structure, this basket weave pattern that we have in the corneal stroma that gives the tissue its transparency. Between these fibril layers, what we have are cells known as keratocytes, and these are what produce this highly aligned collagen. So where we see loss of this structure through disease or through injury, this results in loss of vision. So in order to begin to build our corneal stromal equivalent, we need to have a large, reliable supply of cells. So which cells do we use? The stroma has different populations of cells, but more recently a population known as corneal stromal stem cells have been identified, and these are the progenitor cells of keratocytes. So keratocytes produce the highly aligned collagen that is so key to giving the tissue its transparency in order to aid its function. And the stem cells are highly proliferative, so they're much easier to culture and grow than the keratocytes. So these are the cells that we use for building our tissue equivalent. So how do we do that? Well, we take healthy donor corneas and we digest them with enzymes. We then grow them up in tissue culture flasks in the correct media, so that has the right growth factors and the correct nutrients. And we grow these to large numbers before we then transfer them to a collagen material to further grow our cells. So collagen hydrogels that you can see in this picture here have been used for the last 2 decades to study cells in a 3-dimensional physiological environment. And they're great for studying cells in a dish, as you can see here, but the consistency of these gels are very much similar to jelly that you would have in a trifle, for instance. So when we're talking about making tissues that we can then transplant, these gels suffer from poor mechanical strength. We certainly couldn't transplant it or suture it or fixate it onto somebody's eye. So we needed a way in which to improve the mechanical strength. And the late Professor Robert Brown, who used to work here at UCL, developed this technique known as RAFT, Real Architecture for 3D Tissues. And together with Professor Judy Daniels, they developed these RAFT tissue equivalents for the cornea. And this diagram just shows you a schematic of the process. The collagen hydrogel that I explained is populated with stromal cells, and it's situated between two nylon meshes, and it's placed above this absorbing filter paper. A weight is applied from above, and what this does is it expels 99% of the fluid out into the absorbing paper, and this leaves behind a thin tissue equivalent with our stromal cells trapped inside. But importantly, this increases the mechanical strength and increases the collagen density. And now this tissue equivalent is much more similar to native tissues, and indeed more similar in terms of its strength and its characteristics to the cornea. So this is what the original RAFT research protocol looked like. You can see it's not very reproducible. It's quite messy. Water is expelling in lots of different directions. And it's certainly not suitable for a GMP cell therapy suite. So when we're preparing tissues that would be transplanted to patients, this needs to be prepared in a cell therapy suite that's highly controlled to ensure the safety of that product. So we had to work together with a company called TAP Biosystems, who had a team of biologists and engineers that worked with us to standardize this RAFT process. And this is what it looks like in its current format today. So it went through many different iterations, and now how it looks is we have the hydrogel is cast in a multi-well plate format, like this. And the filter paper has now been replaced with a single porous absorber that absorbs all the water away, and then that can then be discarded. And what you're left behind with is the thin tissue equivalent at the base of the well. And you can then further add cells to it or leave it as it is. And you can see the whole process is very rapid. It takes just under an hour to make. And it's highly tunable, so you can play with the different concentrations of collagen, the thickness of the collagen, which cells you want to put inside. And without working with the company, we would have been unable to do this. And most importantly, the system is now appropriate for clinical use. So it's much more reproducible, it's tunable, but also all of the porous absorbers come individually sterile wrapped, which means we can use them in our clean room environment because everything's much more tightly controlled. So just to summarize, I hope this example of research has shown you how we can combine tissue engineering principles and stem cell technologies to build a corneal tissue equivalent for transplantation. It was a very quick whistle-stop tour of something that's taken the best part of maybe 10 years to achieve. The research has now reached the phase 1 clinical trial, and the first in-human studies are due to be performed. And again, I hope that you've seen through this talk that it's research like this that really requires a multidisciplinary team and an understanding of both the biology and the engineering to tackle a clinical need. And I look forward to taking any more of your questions in the next session. Thank you.